Okay, we're 1 minute after the hour, so we can get started. Good evening and welcome to the Musella Foundation's May Brain Tumor Awareness Month webinar series. Tonight, our special guest speaker is Dr. Pedro Lowenstein. Dr. Lowenstein is a professor of neurosurgery and of cell and developmental biology at the University of Michigan. He's dedicated many years to pioneering a novel gene therapy-mediated immunotherapy for high-grade gliomas, and we're excited to hear about his research. Go ahead.
Great. Thank you very much, Vanessa, for that kind introduction. It's a pleasure to be here today, and I'm going to be talking to you about the new gene therapy for glioma and the results we had in a recent phase 1 clinical trial. So gliomas, as probably most of you know by now, genetically and phenotypically very heterogeneous group of brain tumors. The glioblastomas represent the largest percent of all the primary brain tumors. They form through various mechanisms, including their origin in stem cells, oligodendrocytes, and astrocyte progenitors, and there's still quite active work in this area. The incidence is approximately 7.2 per 100,000, which means around 30,000 new cases in the United States per year. Survival ranges between 6 months to 15 years. Of course, that depends on the malignity of the tumor, the age of the patient, whether they have a tumor with the mutated IDH1 or the wild-type IDH1, and the grade of the tumor. Standard therapy so far includes surgery, radiotherapy, and temozolomide. And what we've tried to do is try to address some of the really problematic aspects of the tumors. What I show you here are 3 modes of MRI on the brain of a patient with a glioblastoma grade 4. And the reason I'm showing you this is at the left, what you see is the brain, and what the arrow is pointing to. Let me see if I just get a quick a laser pointer that you can see. So here, what you see in dark, this is actually the tumor mass. And that is, the surgeon will be able to see that. This is called an T1 weighted MRI after gadolinium contrast enhancement. And now you see the main mass of the tumor, and you see an area in white that we believe is infiltrated by tumor cells. And again, this is visible to the MRI and it's visible to the surgeon. So the surgeon will be able to remove the main tumor, will be able to remove this invasive, first invasive area. However, if you look at it with this FLAIR MRI, you see that you see a huge area here of the brain, which you see in white. That is area that is infiltrated by tumor cells and is also subject to edema, which means water in the tissues of the brain. This whole white area is invisible to the surgeon. And we believe that in this white area, there are the cells that eventually are going to grow back the tumor on the walls of the cavity of the initial resection. So that the surgeon is going to resect this tumor, but he or she can't reach these areas where there are already seedlings of the tumor. So how do you do to target those areas? So we think that the main challenge to induce an immune response against antigens in the brain is twofold. Number one, in order to initiate an immune response, you need dendritic cells, and the brain is very, very poor in dendritic cells. Without dendritic cells, you can't start an immune response. So how will we address this issue? By engineering adenoviruses that express this protein, FLT3 ligand, that differentiates and recruits dendritic cells. Because we inject it into the brain, we believe that it's going to recruit dendritic cells to the brain and therefore be able to kickstart an immune response. And I'm going to show you how our data, both experimentally in animals and in humans, support this contention. The second vector that we use is an adenovirus that expresses the protein HSV1-TK, which together with a prodrug called Valtrex will kill dividing cells. So the combination of one adenovirus that will essentially kill dividing cells and another adenovirus which will recruit dendritic cells to the brain, we believe that we can develop a new treatment for these tumors. This is basically to show you what the viruses look like. There's a protein shell, and you can fit up to 36 kilobases of genomic sequence, and we insert, as shown here down on the lower left, either we insert the gene coding for FLT3 ligand or the gene encoding for HSV1 TK. And these viruses, we can grow them at very high titers, purify them, and inject them directly into what I'm going to show you, into the wall of the cavity of the patients. So what are we trying to do? We're trying to turn a cold immune microenvironment, because essentially in the normal brain tumor microenvironment, there's no inflammation, there's no strong immune response, into what we call a hot glioma microenvironment that is going to be inflammatory and is going to be developing an immune response against the brain tumors. How does our therapy work? The HSV1-TK plus the prodrug Valtrex work in the following way. The HSV1-TK, I show you down here in B, will take the prodrug, which in this case in humans is valacyclovir, and phosphorylates this valacyclovir. The valacyclovir gets phosphorylated a couple of more times, and then it will induce the cell death of dividing cells. So in order for the thymidine kinase plus Valtrex to kill dividing cells, you need the presence of the HSV1-TK protein, you need the access to the prodrug Valtrex, and you need the cell to be dividing. And this will turn out to be important in order to interpret some of the data I'm going to show you in a few slides. How does FLT3 ligand work? FLT3 ligand works on the differentiation from progenitors into plasmacytoid dendritic cells, which are very important in starting the immune response. And it does so for mouse progenitor cells, and for human progenitor cells. So what we predict we're going to see is an increase in plasmacytoid dendritic cells in mice and also in humans. And I'm going to show you the data that we got in humans. So how do we know this works? In experimental animals, we implant, in this case, we implant experimental animals with a tumor. It kills animals in approximately 25 to 30 days. And if you treat it with a number of different controls, all the animals are dead by 25, 30 days. If you treat them with the adenovirus expressing the thymidine kinase, plus the prodrug, in this case ganciclovir, you see a protection of only 20% of the animals. However, if to this group of animals you add the adenovirus FLT3 ligand, now you're protecting 75% of the animals from growth of the tumor. So you're eliminating the tumor growth in the majority of the experimental animals. So this was very exciting, because you see the huge synergy between the thymidine kinase and the adenovirus expressing FLT3 ligand. So I showed you in the previous slide that the therapy really works, but how do we know the therapy is going to work when the patient has a recurrence? i.e., the primary tumor is resected by the surgeon, but the tumor will always come back. And how will our therapy work against this tumor that is recurring? So we did an experimental model for tumor recurrence. So we injected tumor in one side of the brain, and then we treated those tumors with our combined gene therapy. And as I've shown you in the previous slide, 75% of the animals survive long term. So we waited for 2 months to be sure that the first tumor had been completely eliminated, and then we implanted the tumor on the other side of the brain. And when we do that, we are going to test whether we achieved changes in the immune system, so that the immune system now can detect that there's a tumor, it's a new tumor in the brain. So without treating the second tumor, we wait to see what happens, and all the animals actually survive. So not only did we treat the first tumor, but we made modifications in the immune system so that when we challenge now the brain with a tumor on the other side of the brain, the immune system can recognize it and get rid of that second tumor. So I've shown you that probably we've started an immune response against the tumor, but how can we be sure that we indeed started an immune response against the tumor? So in this experiment, we repeated the experiment I just shown you. So we inject tumor on one side of the brain, we treat it with the viral vectors, we wait for 2 months, and then we challenge the animal with tumor on the other side of the brain. But before we do that, we either deplete immune cells from those animals, as you see here in green, or we leave them untreated. If we leave them untreated, just as I showed you before, all the animals survive. However, if I remove the immune cells, specifically CD4 and CD8 T cells, all the animals die. Why? Because we stimulated a memory immune response, which is dependent on CD4 and CD8 T cells. And if I take those away, the animal now dies because I took away the memory immune system that was going to take care of the second tumor. So how does the therapy work? Just as a recap, the virus expressing thymidine kinase will kill some of the cells together with the prodrug Valtrex and expose the microenvironment to antigens and actually innate immune stimulants that are very important for the dendritic cells. And the adenovirus expressing FLT3 ligand will recruit dendritic cells to the brain. The dendritic cells in the brain will be exposed to the antigens, so they can choose the tumor antigens they will stimulate an immune response against. They get stimulated by the innate immune agonist. They go to the lymph nodes. They encounter the naive T cells. They induce the differentiation of T cells, and the differentiated T cells, known as cytotoxic T cells, make their way back to the tumor and hopefully will get rid of the tumor. So this worked, as I've shown you, worked very well in experimental models of brain tumors. So we decided to do a non-randomized open label phase 1, first in human, combined cytotoxic and immune stimulatory treatment of resectable primary malignant glioma. Here are the details of the trial. Patients were consented, but they were only enrolled upon frozen pathology, intraoperative frozen pathology, confirming that this was a high-grade glioma. Once they were enrolled, the dual vector was injected into the walls of the cavity. Surgeon removed the tumor, and then we injected into the walls of the cavity. And they were treated with valacyclovir, the prodrug of the thymidine kinase, for 2 weeks. Then they were treated with standard of care, and at 10 to 12 weeks post surgery, they received a second cycle of valacyclovir. So this was the treatment. It was a simple 3+3 design. The primary objective was whether we to achieve the maximum tolerated dose, and the secondary objective was overall survival. We had 6 cohorts of 3 patients each, and they were treated in a rather conservative manner with the adenovirus expressing TK and the adenovirus expressing FLT3 ligand. We started with lower doses up to a total dose of 2 times 10 to 11 viral particles per patient, half of thymidine kinase adenovirus, half of FLT3 ligand adenovirus. I summarize here the results. We did not reach a dose. We did not reach the dose-limiting toxicity, so the maximum tolerated dose was not reached, and that means that we didn't have toxicity even at the highest doses of the vectors we used, and that means that in the future trials, we can use that highest dose to start to treat the patients in future clinical trials. So that's a very important fact, that we did not have a dose-limiting toxicity. There was nothing specific about the side effects. This here summarizes, let me just move this away if I can. This summarizes the results. The median overall survival was 21.3 months. You can see here that the 12 months overall survival was 72.2%, and the 24 months overall survival was 38.8%. And historical controls will tell you that the 24 months overall survival is 5%. So this compares very good to historical controls. Down below, you see a swimming plot and what happened to each of the patients. And of major interest is that at 2 years, we had 6 patients out of 18 still alive. At 3 years, we had 3 patients still alive, and 1 patient survived just over 5 years. So we were rather pleased with these results, and they compare well with historical comparisons. Okay. All right. So I've shown you that those are the results of the patients. But do the viral vectors have the predicted immune effects in the brain, as we saw in the experimental models? And the answer is yes. If you look down here below on the left, the number of CD8, the total number of CD8 T cells went up, as you see here. The total number of cytotoxic T cells went up, as you see here. And the number of plasmacytoid dendritic cells, the specialized type of dendritic cell to stimulate immune response, also went up. So essentially, the data strongly suggests that we did start an immune response in the brain, and we turned the cold immune microenvironment into a hot, pro-inflammatory, anti-tumoral microenvironment. Sorry, but it's not moving my slides. Let's see if it... Okay. So I showed you that we turned the cold microenvironment into hot microenvironment. Do the viral vectors actually express the transgene? So are the vectors working themselves? And here you see the levels of FLT3 ligand at 2 weeks compared to pre-surgery values goes up. So FLT3 ligand 1 virus is functioning. And what you see here on the right is immunocytochemistry for HS3-1 thymidine kinase immunoreactivity. So we can see the actual gene encoded by the virus up to 17 months post-viral injection into the resection cavity. So the viruses are working, and the viruses are doing the changes that we wanted them to implement in the brain. So this was published last year in the Lancet Oncology. This is the title of the paper, Combined Cytotoxic and Immune Stimulatory Gene Therapy for Primary Adult High-Grade Glioma, Phase 1 First-in-Human Trial. These are the 31 authors, and everyone who contributed, from the neurosurgeons to the neuro-oncologists, to the radiologists, to the pathologist. We were all at the University of Michigan, so an enormous group effort, and a number of people who were in the lab that did all the correlative studies, and everyone is cited here. To summarize the findings from the trial, were that the dual vector treatment is safe and potentially effective. The maximum tolerated dose was not reached. Both transgenes are active within the peritumoral brain area. The tumor microenvironment becomes activated, and the transgene HSV1-TK is present up to 29 months post-delivery into the brain. So what future trials are we planning? We're planning to extend the period of administration of Valtrex because of the longevity of the HSV1-TK in the brain. We want to potentiate the number of cells that could be killed, tumor cells that could be killed by this mechanism. And we are combining our approach with various immune modulators. We're doing future trials with combining the gene therapy with checkpoint inhibitors, with various inhibitors of myeloid-derived suppressor cells. And also we're starting a trial in combination with GCSF. And finally, a big thank to the patients and their families for their trust in participating in the clinical trial. Basically, I am acknowledging the people in the lab who did all the correlative work, and funding, which we had mainly from the Phase 1 Foundation and the Board of Governors from Los Angeles. So I think this is the end. Thank you very much.
Thank
And I leave
you.
you with this slide.
Thank you very much, Dr. Lowenstein. We do have some questions. The first one was, the patients in this study all had either a gross total resection or a near gross total resection, correct?
Correct, yes.
Okay. Do you feel that that played a part in the positive survival results? And would you even be able to use this approach in someone who still had considerable residual tumor?
Yeah, so that's a very interesting question. It was decided that this would be one of the entry criteria in the trial, and in the way you design the entry criteria to be safest for the patients with an early Phase 1 trial. I think it would be very interesting to move the trial to treat patients where that are not amenable to gross total resection, but to partial resection, because I think that will give us a window into, now we have some tumor left, and if we inject those areas of the tumor, would that give us an even better immune response? So I think it would be a challenge, but would be very interesting to plan a new trial with that population of patients.
You mentioned that you didn't hit the max tolerated dose. Was there an association between the higher doses and longer survival?
No, we tried, we looked for a number of correlations, but there was no correlation with survival for the highest doses. Although if you look carefully into some of the correlative studies, the highest doses had possibly highest secretion of the cytokine, and in the HSV1 TK, the number of patients is very, very low to make those kind of assessments. And I think that in future trials, we'll start with what was the highest dose here, and that dose will be accepted by the FDA because we didn't hit the MTD.
And you have for the future trials, you have potentially extending that period of Valtrex administration. Would you also then, re-challenge with injecting more of the adenovirus?
Ah, okay, another another interesting question. So by the time we planned this trial, the consensus in the field was that you would inject patients once. It now has been shown in a trial done in Japan that they used a different virus. They used an oncolytic virus, and they did up to 6 different injections, 6 open surgeries with 6 injections of the oncolytic virus, and it was relatively well tolerated, and it looks like they might have had interesting efficiencies as well. So that's a possibility. Starting with the more conservative aspect, the interesting thing about the decay is that you need the decay, you need the prodrug, and you need the cell to divide. So we've shown that when we detect the decay up to more than 2.5 years in the brains of the patients, we still see them in tumor cells. So the tumor cells that are dividing very slowly. So that's why we think that extending the Valtrex, we could really therapeutically hit those cells. But the multiple injection is something to consider as well. So it's a very interesting proposal.
Okay. And there were some studies in Sweden on valganciclovir as an adjuvant to standard of care. Are you familiar with those? Do you have any opinion on those just as a standalone adjuvant?
Yes, I can give you my opinion. Um, I don't think that they've shown that the drug really has an effect. It's a bit of a statistical I don't know how to call it. It's a statistical trick that allowed them to say that, because basically what they were saying is that patients that take more Valtrex live longer. Actually, what they were doing, they were treating the patients and they were giving Valtrex all along. And of course, the patients that survived longer took more Valtrex. But there's no causal relationship. They also drank more coffee and
Yeah,
drank more Coca-Cola. And
the immortal time bias, yeah.
yeah, yeah. Oh, that has a name, that fire. But that's, yeah,
Okay.
yeah.
Um,
So I don't, I, I think it's interesting. What's interesting about the Swedish study that it supports us in treating patients long time with Valtrex, but I would do it after we inject the vectors, definitely.
okay. And another question we got was how far out from the area of injection do you see the transgenes active? So when patients had recurrence, was it within the area that the virus, had been injected?
Right. Very, very observant question. I would need a whiteboard to um to answer this question. But basically, I guess you can see my playing with my hands here. So if this is the resection cavity, the virus was injected into the resection cavity, and I think that it can probably diffuse 1 to 2 centimeters into this area. Okay. Interestingly, the tumor 80%, 90% of the time comes back in this sliver of tissue of 1 to 2 centimeters surrounding the tumor. And I think we have now evidence that proves that assertion, because we injected the virus here, the tumor comes back many months later, a surgeon takes it out, and we can still see the expression of the thymidine kinase. So I think the tumors coming back in the area, that area that I showed in the first slide was invisible to the surgeon, so they can't resect it, but it's where the cells are that are going to come back and reconstitute the tumor.
Okay. And I think I read in the paper that, or maybe in an article about how this therapy might be more effective in IDH mutated gliomas. Can you speak for a minute about that?
Yes, in IDH1 mutated tumors, Maria Castro has shown that they have the immune cells in the microenvironment of the IDH1 mutated tumors is less inhibitory than in wild-type tumors. And therefore, these therapies may have a stronger effect. And they do in experimental models, they have a stronger effect in the IDH1 mutated tumors. So yes, that might be. In our trial, we had no IDH1 mutated tumors.
Yeah, and still that almost 40% 2-year survival, which is impressive. Do you think that adding, things like the GCSF would help more for the wild type immune response in that respect?
Sure. GCSF turns out to be one of these molecules that biases the immune system in the mutated IDH1 tumors. So we found that experimentally, and we would like to test that. And the idea there is to make that the tumor immune microenvironment less inhibitory of immune responses and more viable to carry the immune responses against the tumors.
Okay. And can you talk, so the phase 1 is completed. Are there any open trials now?
No.
No, okay.
Now,
When do you
hopefully,
expect to, yeah,
well, hopefully we want to start one trial pretty soon
okay.
with some initial patients. But I think it may be probably at least 3 to 6 months until we can start treating a larger number of patients.
And would that trial only be at University of Michigan, or do you envision that being multi-center?
No, we are now collaborating with a number of centers. So the first trial will be in combination with a group at UCI, University of California, Irvine, and we have other collaborations planned with other groups throughout the country. So they will all be multi-center trials.
Okay, great. It's good to hear about these exciting new promising therapies, but the patients always want to know,
Sure, of course.
how can we access them?
Sure, and
That's
yeah,
great.
if they want to write to me or find out more, I'll be happy to talk to them.
Okay, yep, maybe you could share your email with Al and I, and when we share the recording, we'll put the contact information in there as well.
Sounds great.
Okay, I think that was all of our questions. Thank you so much, Dr. Lowenstein. This is a great presentation, and thanks for answering all of our questions.